We're going to continue with the concept of convolutional neural networks. And just take a good look at this because it all began with cats. Now, the story so far, I can't guarantee that I have all the equations on the slides because these have been printed into PDF and sometimes things break. The good news is there's nobody in class, so nobody will know I made mistakes. The story so far, we've seen that pattern classification tasks such as does this picture contain a flower can be performed by scanning for the target pattern. And scanning with a network and combining the outcomes is equivalent to scanning with the individual neurons in a sequential or a hierarchical manner. The outcome is going to be the same. You would get one decision per position. You'd be voting over the lot for the final outcome. We saw that the deformations and the input can be handled by max pooling. The 2D case gave us a convolutional network, also called a ConNet. If you're scanning only a long time, for example, if you're looking at speech recordings and saying, does this recording have the word welcome? That was called a time delay neural network, but the concept was exactly the same. So where did all of this begin? Where did that crazy little model come from? And it turns out that a lot of the components that we saw yesterday were not motivated by pattern classification. In fact, some of them are completely can be completely eliminated, so at least some of the components. So to get an idea of where it begins, we have to consider a little bit of history. How do animals see? When you build a convolutional neural network to analyze pictures, what you're basically trying to do is to make the computer see. And of course, like everything else in neural networks, it starts with this question of trying to analyze the processes of the human or the animal brain when they actually see pictures. Now, in the early days, the analysis was based of when people try to understand human or animal vision, the research focused on behavioral response. How do people respond when they are shown some images? Which led to study of visual illusions, things like Gestalt. So here are some examples of Gestalt. You will note that you don't really have a panda over there, you just have some black patches. But then you see the panda because your brain fills in. There really isn't a cube over there, they're just 6 versions of the peace sign, but somehow you're managing to see a cube. So this is your brain filling in. All of this has to do with behavioral response, how you interpret things. But there wasn't any insight into what was happening inside the brain at the neural level. That work started with these two guys, Hubel and Wiesel, in 1959. This was either the very first study or one of the first studies on neural correlates of vision, definitely the most famous one. So there was this study on the receptive fields in the cat striate cortex. What is the striate cortex? Turns out that Mike Tarr might have gone over this. When you actually see something, the signals from your brain first go to the back of your head, something called the V1, and then they go through several layers of your brain, V1, V2, V3, until you actually make your interpretation. Now, the V1 in cats, the equivalent of the V1 in the brain actually has a striated structure, so they call it the striate cortex. It's basically the V1. These two guys studied the neurons in the V1 layer. And they did this using 24 cats. That's why the cats, which were anesthetized, immobilized, they put on artificial respirators, they used truth serum to anesthetize them. And then they did something, that was just the beginning of the stuff they did. And then they stuck electrodes into the cat's brain. Nowhere in the paper do they say they actually killed the cat, but at the end of the paper they say the brain tissue was studied. So I'm assuming something bad happened to the cats. Anyway, here's what they did. They kept the cat was anesthetized. They kept the iris open. They kept the eyes open, and when the cat is anesthetized, the iris kind of opens up naturally. And then they beamed light of different wavelengths along different patterns into the cat's eye and studied the response of individual neurons at the back of the head and the striated cortex. Now, what they found was, first, something that's obvious. Vision starts with the retina. your retina is one of those crazy organs in your body which proves there's no God, because they can't be a designer designing something so stupid. It starts off with neurons. The retina actually has a layer of light sensors, but crazily enough, these light sensors connect to neurons that are not behind them. The neurons are actually in front of the retina. Now, which means that these neurons have to fold back and go into the brain through a hole in the retina, which means that you actually have a hole in the retina out here through which all of these neurons go out. And so that's why you have a blind spot in the eye. There's this spot in the retina through which the light is going back, the signals are going back into the brain. Now, the first thing you find is that any particular spot in the retina actually responds to light. But then if you look at what happens to, now if I think of the retina as a circle, and this is the brain, any cell in the striated cortex actually responds to not just one cell in the retina, one position, but to an entire range of positions. So this was called the region of the image that this responded to was called the receptive field. This is a terminology we will use even in our new ANNs. This is called the receptive field of the cell. The receptive field of the cell had an interesting property. Light that fell outside, the neuron didn't really respond. But within the receptive field, you had areas. So let's say this is the receptive field. You had areas where if the light fell on this area, the neuron responded. You had areas where if the light fell on these areas, regardless of what happened on the this region, the neuron would fail to respond. So these were the excitatory responses, these were the inhibitory responses. And so if you actually had light, diffuse light falling over the entire receptive field, the neuron really wouldn't show any extraordinary response. Now, the neuron only responds if there is light in the regions where it's looking, and there is no light in the regions where it isn't, where it doesn't want to sense light. That's how it makes patterns. So what these guys did was to wave this little pinpoint of light of different wavelengths in different directions. And consider what happens over here. If I have something of this kind where this is a plus and these two are minuses, if my light is oscillating in this direction, the pluses and minuses will cancel out. If my light is oscillating in this direction, then it's going to stay much longer in the plus region than it is in the minus region. So you're going to begin seeing more and more excitation. If the light is waving exactly up and down this way, it stays in the excitatory region, so you're going to get a lot of response from the neuron. So this is so here's what you would get. What they found was that these neurons responded to patterns, linear patterns, oriented in specific directions. So they were not always vertical or horizontal, but each neuron had one specific kind of pattern that it was actually responding to. And so here's one of the figures from their paper. In this case, the left and right, they just show where one is where they're going across the entire receptive field. The second is where they're going over half the receptive field. And you can see that this is a particular neuron that responds best to vertical lines and not to horizontal lines. So as the angle of the excitation of the retina changes, the pulse trains become denser and denser till they're actually showing a light in an exactly vertical manner. Now, this neuron is sensitive to vertical bands. So what they found was that oriented slits of light were most effective stimuli for activating striate cortex neurons. And later they show that you actually get two levels. Here you're speaking of a single neuron. But what they showed was that there are two levels of processing. And they call these two levels of processing S cells and C cells. Now, when you have something of this kind, and light falls on the retina, what really happens? Now, for an S cell to actually have a response like this on the retina itself, each point is going to go. Each point responds to a circle of light around it. So the response of any given neural cell, simply because of scattering, is going to be somewhat circular. So what the S cells were doing was to connect to linear arrangements of these retinal cells, and when all of them fired, they were firing. But then, because of the inhibitory and excitatory regions, sometimes they wouldn't fire. So which means that if you had fuzzy patterns, if you had noise speckles, these S cells wouldn't fire. Now, so you had the higher level, which was the so-called C cell, which looks at multiple S cells at the same time. So if you have some region in the retina, and you have one, you're looking for this kind of a receptive field where you're trying to look for a vertical slit, vertical line, there would be multiple S cells looking at that same location, and the C cell was simply taking looking at all of these S cells which are looking at the same region. And if any of them fired, it would fire. So it was they were just picking up the maximum signal. This sort of cleans up the response. We want to be now, again, observe that it's not filtering, it's picking up the largest signal. That's because as human beings, as animals that must survive, it is important for us to respond to light. We are not in the business of, the eye is not in the business of denoising, the brain is, right? So it sort of makes sure that the brain responds. So, but then as you go through the brain, they also found that there was a hierarchical structure. You had these S cells which were detecting patterns. You had C cells which were looking at groups of S cells to make sure that they actually picked up signals. But then the outputs of these C cells were looked at by the next group of S cells, which were looking at patterns in the outputs of the C cells, and those in turn fed to, and the next layer of S cells fed into more C cells, and so on. And as you went deeper and deeper, the patterns that the C cells were looking for became more and more complex. They were no longer just lines, but there were combinations of lines and other such stuff, much in the manner that we spoke of when we were speaking of how neural networks themselves build up patterns. So, and their further studies actually worked on monkeys, and things got really bad, so I won't go there. And of course, to add insult to injury, it turns out that maybe the work wasn't particularly thorough, right? And those poor cats, well, anyway, won't go there. But so you actually have the first real model of how neurons respond to input images. But this was in 1959. In 1980, this guy, Kunihiko Fukushima, decided to computationalize it. First, he found that Hubel and Wiesel's work had some deficiencies. For example, there's position invariance in your eye. Now, you have something called a grandmother cell in your brain. Have you heard of the Jennifer Aniston cell neuron? I'm pretty sure some of you have heard of the Jennifer Aniston neuron. So guys who work on these kinds of things really like epileptic patients, because when you have epilepsy, you go to the doctor, the guy opens up your skull and leaves you there with your brain exposed for a while, meaning several days. So these guys will go and put these little sheets of sensors on your exposed brain and run experiments on you, because after all, you're a captive audience at this point, right? And they caught one of these guys and began showing this person various images, and they found that this specific character had a neuron that fired only when he was shown pictures of Jennifer Aniston. So this is the famous Jennifer Aniston neuron. There was a paper in Nature about it, if I recall. There's nothing special about Jennifer Aniston. He probably had one for every single actress and every single person that he recognized, including his grandmother. So we call these the grandmother cells. And the grandmother cell will fire regardless of whether your grandmother is here or here or here. This position invariance. And this simple C cell S cell combination that Hubel and Wiesel spoke about were tracking individual neurons. They were not actually accounting for the position invariance, that the same kinds of patterns must be detected regardless of where they occur in the image. So he came up with a nice little computational model. He said that the visual field actually has a series of a hierarchy of modules where within each module you had an S cell C cell combination. Remember, without the C cells, the S cells aren't really doing very much because the S cells are kind of by individually not good enough. So the C cell is voting on them or picking up the largest of these to make sure it captures all of the signals, right? So you had many such layers. First, the images on the retina would go to the first layer of S cells. The output of the first layer of S cells would go to the first layer of C cells. So there's a one S cell C cell block. The output of those C cells would go to the next layer of S cells, which would feed onto C cells, and so on, all the way until the end. Who knows what the end is? Now here's how. But then you want position invariance. And so here's how he hypothesized the system would be. And there's reason to believe that his hypothesis is kind of correct. That let's consider your grandmother, but specifically let's say your grandmother is a cat, and let's specifically consider the eye in your grandmother's eye, which has an iris, which is vertical, right? So now in that case, if regardless of whether granny is here or granny is here, you're going to be looking for her eye, correct? So you want to have this granny vertical eye slit detector regardless of where granny is in the picture. So, but then the neurons in your brain are not connected to the entire range. Each of them is locally connected. So the only way you can account for this is that if you have, assuming this is your retina, if you have an entire plane of granny eye detectors where you have many neurons, each of which is looking for exactly the same pattern. So between this plane, it's covering the entire input image, but each of these guys is looking over only one location in the image. That would be a granny eye detector. But then your granny has other features, right? If your granny is old enough, she has a mustache. Then at which point she's going to have something horizontal. And so you're going to have a granny mustache detector, which is doing exactly the same thing, right? Which is something like that. So he hypothesized that there are within each SC combination, you have several such S planes, where within each S plane you have a number of neurons, each of which is looking at a different location in the input image, but all of them have exactly the same response, right? And now, of course, you need a C cell layer which cleans this up. The C cell layer is only looking at the output of the S cell layer. So you're really making sure that you're seeing granny's vertical eye and that you're really seeing granny's mustache, okay? And then you have the next layer, which is looking for granny, and now this is an S cell. But then how do you put granny together? You got to put together her eye, you got to put together her mouth, her lips, every single thing, right? So this is going to be looking, any granny cell over here, which is looking for some component of granny, is going to be looking for her eye, it's going to be looking for her mustache, it's going to be looking for whatever her nostril, each of these put together. So one S cell in the next layer is looking at every single plane, but these are C planes. These are all S planes. These are C planes. This is within one block. The C has cleaned up the S, right? And then over here you would have a bunch of these guys, each of which is going to be looking at an entire collection, right? And these are S's again, and then they'd be followed by C's. So observe that a C plane looks at one S plane, an S plane looks at the entire collection of C planes from the previous layer, right? Makes sense. And now, specifically in Fukushima's model, these guys were looking at the receptive field of each guy was actually elliptical. So this one would be looking at an elliptical region. The corresponding C plane is going to be looking at an elliptical region out here to decide if there's really a vertical slit, right? And then this guy is going to be looking at exactly corresponding elliptical planes on every one of these planes to see if the specific combination of features occur. And then this guy is going to be looking at an elliptical region out here. So all of the receptive fields for every single neuron was elliptical. But then if you look at the receptive field for this neuron over here, this is an ellipse. Each location here comes from an ellipse over here. So by the time you work your way out here, the receptive field for the second layer is going to be bigger than the receptive field for the first layer. And as you go deeper and deeper, the receptive field for any single layer neuron is going to get bigger and bigger and bigger, right? And now he also had formulae for when each of these neurons actually responds. So this neuron, for example, is looking at an elliptical field, so it's obviously looking at all of the cells in that region, but it's combining their signals in some manner to decide when to fire. And the formula he came up with is that ugly formula on the slide, which I won't read out because it's not worth the trouble. It turns out it looks like a ReLU. Then in the C plane is looking over at an elliptical region of pixels in the or neural responses in the corresponding S plane, each cell in the C plane. So again, it's combining the whole lot. It's combining them using this rather hideous formula, but it turns out the hideous formula looks a whole lot like a max. And so you can collapse the whole thing into layers of S planes where each cell has a ReLU-like response to the collection of inputs, and the C cells, which actually look at, have a max response to the elliptical region in the S cell output. And so you could simply replace this with ReLUs and maxes and you'd be just fine, right? Now, the deeper the layer, the larger the receptive field of each neuron. And something interesting also happens, because if when a S cell within each S cell C cell layer, so let's say you have an S plane, and in the C plane, if you've got one cell over here and it's looking in this region for vertical slits which correspond to your grandmother's eye, then this region of neurons really corresponds to vertical slits, high vertical slits, right? So the next guy over here, it's looking at a slightly shifted region, which could perhaps even be overlapping like so, for vertical shifts in a slightly shifted region, but there's a significant shift. So as a result, this plane actually ends up becoming much smaller than the S plane. Then the next layer's S plane is going to be more or less the same layer as the C plane, but the second layers, so the next S would be the same size, but then the corresponding C is going to be smaller. So as you go deeper through the network, all of the planes get smaller, right? This is just the architecture as originally proposed by Fukushima. You need a learning mechanism. The learning mechanism is, how do you know what is the response? Now see this guy, right? This guy, if this is no. Let's look at the S plane, right? This S plane is looking at a region within the C plane, and it's combining all of the responses within that region to decide if it has found a feature or not. So it needs to have weights to each location over here. How exactly do you decide what weight to apply, right? Now this is where this business comes in. Even within so if any particular input looks at detects a specific feature, if any particular cell detects a feature, then the region of the image that contributes to that feature cannot be contributing to any other feature, right? So What this meant was something of this kind. Let's say I had this collection of S-plane images, right? And then C-plane images. These have been already captured and filtered. And then I have the next set of S-plane images, S. So this guy is looking here and here and here, same location, and so on. But so is this guy. Correct? They're all looking at the same location. Now, if this corresponds to a mustache, it cannot correspond to an eye. So which means that if you decide that this guy has contributed to this location here, it has definitely not contributed to this location. So the way they trained this network was to initialize all of the weights randomly, then provide it with inputs. Then they would find out in response to each guy which one had the largest response. So this could have responded, or this could have responded, or this could have responded, right? Which one had the largest response? They choose only one and attribute this pixel to only the largest guy, and then use Hebbian learning to update this weight, which of course will reflect for every neuron in that particular map. So very simple Hebbian learning growth. You can computationalize it. We won't get into the details, but this winner take all ended up giving them this kind of beautiful model where they experiment where they showed lots and lots of different pictures to the network. And they found, here's what they found. They found that, for example, if you showed lots of characters, the early layer S-planes would begin to respond to different components of the characters. So maybe one S-plane would correspond to this stroke of an A, another to the horizontal stroke of an A. And then the later ones would begin putting these together. All of this is unsupervised. There are no labels. You're just presenting inputs and using Hebbian learning to decide which ones will fire and which ones to update and which ones you shouldn't. And eventually they ended up with results like this, where they showed that if you just give it lots of pictures, the responses in the final C layer actually automatically learn to cluster concepts. So for example, you would find that one of the C cells responds in response to the digit 0, regardless of whether the digit 0 was noisy or not. The reason it was able to respond to noisy inputs was because of this filtering that the C did to the S-plane responses, right? Now the key point is all of this is a computational model of a biological model, right? The only thing happening over here is that the entire thing is unsupervised. What can you do to make this one step further, or take this one step further? Go beyond Hebbian learning and actually add some supervision. Instead of saying, decide, just learn what you're going to represent. I want to say this guy, I want this to respond to the character A. Very simple. Now two things over here. Observe that only the S-plane weights are learned. S-planes are plastic. C-plane weights are not learned. They are just picking the max within the receptive field. So you only have to learn the S-plane responses, right? So here's what you would do. You add external supervision. There were many models for external supervision. The earliest one was actually by Homa and Atlas in 1988. Then Kevin Lang here in CMU figured out that you could add similar responses when you're analyzing time series data, particularly speech, and that was the time delay neural network, 1989. And Jan LeCun, a little later, or more or less or about the same time, applied this to analysis of images. So there were multiple people who came up with the same idea at approximately the same time. But the most successful guy, of course, was LeCun, because he applied this for images and it took off. But here's what they did. At the end of that final layer of C cells, they just added, you take all of these C outputs and put it through a softmax or a logistic. And now you get a binary output, right? And then you can actually present inputs and outputs. You can present pictures of 0 and call it a 0, pictures of 1 and call it a 1. And define a loss function, backpropagate the loss function, and learn all the weights, right? And these are shared parameter networks. This is exactly completely learnable using backpropagation. The model is well defined. Again, you only have to learn the S cell parameters, not the C cell, because the C cell is fixed. The S cell is the only one that's plastic. Now the thing is, computationally you can make a few distinctions. So in any S cell, if I have each position, each neuron looking at different regions of the previous C cell with exactly the same response, this is exactly the same as scanning, right? Because they're all the same neuron. So instead of implementing it as a block of many identical copies of the same neuron, you can have a single neuron scan the image, which is what LeCun actually did in his implementation. So this another thing you did for computational purposes, elliptical receptive fields are hideous. So he replaced them with rectangular receptive fields. But that doesn't really change anything, right? And so you have to define the activation functions, and you have to define and the responses for the S field and the C S planes and the C planes. Now the S plane, look at what the S plane is doing. The S plane, each neuron in the S plane is looking at the outputs of the previous C planes. But since there are multiple C planes, as we saw, right, it sums over all of the C planes. Within each C plane, it's going to look at a rectangular or a square region. And for each position in the square region, it has a different weight. So this weight itself has 2 arguments within any plane and changes by plane. W has 3 arguments. So you would have, if this guy is looking over a whole collection of these planes, within each of these, it's looking at the same rectangular region. There's going to be a bunch of weights here. So this is a 2-dimensional variable and a bunch of planes. That adds the 3rd dimension, right? The activation function sigma in the original work, he just used a sigmoid. And for the C plane, of course, all you're really doing is looking at a rectangular region and just picking up the maximum activation. No big deal, right? So problem solved. Now, and here is this, I don't know if this works. Okay, this is a PDF, but if you actually saw if I had the ability to show the PPT, you would see that he actually created this demo, the system that could recognize handwritten digits. And that's why this thing became really famous, because that got taken up by the US Postal Service to scan letters, addresses and letters, specifically the zip codes, and that got used a whole lot. So it actually immediately went into practical use. Anyway, so you get the idea. Yes?
So I have a question about the fact that when you go from C to, you go from one layer to another, and you have a neuron that recognizes the eye versus a mustache, then this neuron should be used only once, right? The one you said that if you use one of the connections, you want to drop the others.
So
not in the CNN, but in that natural model.
In the Fukushima's model, right?
Right. So that doesn't sound natural, because sometimes when you recognize just a small piece of rectangle, it can actually correspond, say, to an eye in a grandma, or to some different thing in a dog, and then.
It certainly can, right? It turns out this is just a model. And it turns out that if you're presenting grandma and you're presenting an eye, right? So when you're presenting a grandma picture, then this one has the highest response for this, in response to this guy, and you update this weight, okay? But then when you present a dog picture, the same guy, this one ends up having a higher response. So that weight gets learned as well. So you actually end up learning complex patterns. You're just picking which one to update using Hebbian learning. It's just a model, right? So, oh, the question, he just wanted to ask, the question was, if you're always picking the max, then you're assigning one input to one feature, whereas an input can correspond to multiple features. Like the corner pixel can correspond to both the horizontal line and the vertical line of a square. So wouldn't this learning be incomplete? And the answer is no. It actually would end up learning both eventually, right? So here's the model. Here's the story so far. The mammalian visual cortex consists of S cells, which capture oriented visual patterns, and C cells, which perform a majority vote. Fukushima's model, which is called the neocognitron, emulates this behavior with planar banks of S and C cells with identical response. And Jan LeCun's LayNet added external supervision to the neocognitron to actually learn this from labeled data as opposed to unlabeled data. Questions? From here, we're really going to speed up. Here's the general architecture of the convolutional neural network. Oh, good God. I was kind of counting on having PowerPoints. I have several animations over here, but let's go through this anyhow, right? This lecture may leak over, in which case I will record something and you're going to have to watch it. We finally figured out how to do it. So anyway, a convolutional neural network comprises convolutional and downsampling layers. The S layers are the convolutional layers. The scanning of an image, we will call it a convolution. The C layers, which actually take max votes, end up reducing the size of the S maps. These are the downsampling layers. And there's no need for every S layer to be followed by a C layer. You can have any number of S layers and you can have before a C layer occurs. The original model had one C layer after every S layer, but the downsampling can happen at any position in the sequence. And once you have got through all of these, the final downsample layer can be put through a single neuron or in fact an entire multi-layer perceptron, just so that you can actually perform pattern classification on it. This is the convolutional neural network. Now, so convolutional neural networks are learnable. All of the convolution layers are learnable. The downsampling layers are not. So downsampling layers are generally fixed and they're not learnable. The convolution layer itself corresponds to a series of maps corresponding to the S planes in the neocognitron. We might also call them feature maps or activation maps. And the convolution layer, each plane in the convolution layer actually has two subcomponents. The first is this, that within this convolution layer, you have this guy scanning the input. When it scans the input, it's actually computing a weighted combination of the inputs in the previous layer. But then that's the first round. The second round, every one of these things is then put through an activation function. So although I've represented it as one layer, it's useful to think of it as two distinct actions. One is the scanning, and the second is the application of the activation, right? In computing any particular layer, every single map in the previous layer contributes, as we just saw, right? If you're recognizing grandma, you want to find her eye, you want to find her mouth, you want to find her ear, you want to find her hair. Every one of these guys must actually contribute into every position in the S maps, right? In the convolutions. Now, exactly how does the convolution of a single happen within a single layer? Now, this was supposed to be a magical animation. That's not going to be an animation anymore. So imagine I have a 5x5 image with binary pixels, like so. And now, here is something, a bit of jargon. When this one looks at a region within the previous image, right, previous plane, it's actually taking a weighted combination of all of the values in this region. The set of weights is what we will call a filter. And this again derives from signal processing literature. So you're going to have the set of weights is going to be this square times as many planes as you have in the previous layer. That entire collection of weights is what we will call a filter, right? And so, but these are just the weights. Now consider what happens to one of these guys, and let's just assume that you have the 3x3 filter. Now this is very unfortunate that I don't actually have the animation. So we're going to hope that I can explain it without animation and you just get it. So here's what would happen. You would place that filter on the top corner of the image, top left corner, and the filter values are shown in red, the image value is shown in black. So you do a component-wise multiply of every pixel value with the corresponding filter value, and then you'd sum them up. And you see that if you sum them up, this particular sum is going to be 4. Then that entire filter is going to be shifted by 1 pixel, and you repeat the same operation, and then you get the next value. Then the entire thing is going to be shifted by another pixel, and then you get the next value. Then you come back to the bottom, you slide the thing down 1 pixel, you're going to get 1 value. Then you slide it right 1 pixel, you get a value, and so on. Eventually you end up scanning the input, right? Now, now the stride between adjacent positions, you don't need to necessarily just skip by 1 pixel. You could be skipping by 2. So here, for example, I could be just placing it here and getting 1 response, and then shifting by 2, and I compute the next value. Then I shift down by 2 and get the next value, and I shift down by 2 and get the next value, in which case the size of the result is going to be halved, right? Or any number of, you can stride by any number of values. Now what really happens, this is key, I showed this for one plane, but there isn't actually one plane. So if you look at any particular single S-plane over here, right, each filter actually looks like this. You're going to have one square patch or one rectangular patch for every plane in the previous layer, right? And so you would have, you see the number of squares over here. The receptive field size is 3 cross 3, but the number of parameters is 9 times p, where p is the number of layers, the number of planes. So here I'm showing 3 planes, but there are triple dots, so they're all at least 3 planes. The number of parameters in a single filter is at least 27, right? And the actual computation is like so. You're computing, so the filter actually has now, this is the layer number because you need to index the layer as well. This is the plane, and these two are the position within the square within the plane. And in the previous, and what you're doing is you're taking a square region within the plane, you're doing a pointwise multiply, and you're summing over the entire square for each plane and across all of the planes, right? That's basically what this operation is performing. And so that is the z value. So here's how you would actually compute. You would slide forward 1 position at a time, and at each position you would be computing this total value. Observe that the summation has 3 terms, right? 2 axes, 2 indices are for the x and y span of the filter itself. The third index is across all of the planes, and you'd compute the entire figure. So for the second, now this is for just one plane, right? You're going to have many such planes over here. So the second plane is going to have its own filter, which could be completely different. And the second plane is going to be performing its computation pretty much the same manner, going left to right, 1 pixel at a time, and performing this computation. There's a different way of viewing it. Instead of drawing all of these planes top to bottom, a much more convenient way is to say they're all just they just form one 3-dimensional cube. So if I think of this as forming one 3-dimensional cube, my filter doesn't comprise many independent square patches. My filter itself ends up being a cube which consists of a stacking of many of these square patches, right? And now I can view the computation like so. I place the filter, which is a cuboid, on this cube which represents the stacking of all of the planes. Now I'm just doing a pointwise multiply and adding the whole lot. So now it's a 3-dimensional sum, as you can see. And this would subsequently, so the sum actually has these 2 parts. The first over here, that is the sum within a single plane as shown by the highlighted yellow region. The overall sum is across all the planes as shown by the collection of highlighted yellow regions, right? And the actual computation is going to slide left to right in that manner to get the entire picture, right? Entire computation, yes?
So for that filter box or that stack filter, are they the same filters or are they different filters?
They're all different, like we said, right? So that stacked filter is just this guy, this image here. And look at the color coding tells you they're all different in each plane, right? So see the pattern of colors. I'm just explicitly indicating that they're all different numbers. Right? You have a different, you're looking for different patterns. The weights you would use to look for the eye are going to be very different from the weights you would use to look for the mustache. Right? So obviously they're going to be different.
And we're summing them together?
Yeah, you're going to say, I need to see an eye, I need to see a mustache. Total evidence, right? Yeah. Yeah.
You go to the next.
This.
So that like filter one, that's using the same like weights for each dot.
So this guy, so yeah, it's the same set of weights for every dot on the plane, right? You're just taking this cube and you're scanning. So obviously it's always the same set of weights. You're just scanning, right? Was that your question?
Yeah.
Okay.
This sounds like you didn't say that.
No, okay. No, when you're saying of different weights, you're saying for each plane there are different weights. So for every yellow patch over there, each yellow patch represents a different set of weights. So this cube is a whole collection of different weights, and that is scanning the entire image, right? Yes?
So if you're doing downsampling, why you need a bias? Because you found the max, so there's always.
Pardon me?
Because you're doing the max.
But you're retaining the max value, which will keep the bias.
Yeah, but after that you still make max, and you're adding a value in one of the regions. So
Let's hang on to it for a couple of slides, okay? Then you ask me this question. So currently we are still speaking of the convolutional layer. We haven't yet gotten to the downsampling, right? So, so here's a piece of code, pseudocode. You can see what's happening. I'm going over all the layers. You're going over each neuron, which basically represents a filter, right? One of these cubes. And each cube is going over a patch of the input. But then you take the collection of patches from all of the planes. So this would give you a 3D tensor. And then there's just a component-wise inner product between the weights, which are a cube, a 3D tensor, and the segment that you pulled out. And that's the sum that you're going to be storing in Z. Okay? That's giving you the plane. Now there's a two-step process, as we saw. Once you compute all of these values, these guys are, so before I continue, yes. Observe that the stride may not be 1 pixel, but there's something else that also happens. The size of the output is going to be different from the size of the input. Why so? The filter is 3 pixels wide over here, right? 3 elements wide. So I can only slide forward 3 times before I run out of picture. If the filter is n elements wide, I'm going to lose n minus 1 columns and n minus 1 rows. Because after that I won't have enough pixels in the image to actually accommodate the filter. So every time I scan, my image is going to shrink in size. That is with a stride of 1. Now if I have, so here is what, now this was the animation, lovely animation that was going to show you this response. Now if I have a stride of 2, what would happen? If I have a stride of 2, the size is going to shrink even more, right? You'd basically get an output of the same size as the stride, we'd get with a stride of 1 divided by 2 in every dimension. And so that is going to change, right? So in general, if I have a, so here I'd get although the input is 5 and the filter is 3, I subtract what happened. Did something show up? Okay. So I subtract, because the filter size is 3, I have to subtract 3 minus 2, which is 2, from the size of the input. That leaves me with just 3, right? And remember that even if I have to add a plus 1 afterwards, because if I can only fit the filter exactly once, I'm still going to get 1 point, right? So 3 plus 1 is 4 divided by 2. That's going to give me the number of pixels I'll actually get. So in general, if I have an image of size n cross n and a filter of size m cross m, then if I have a stride of 1, the output size is going to be n minus m minus 1, right? So if the input is size n and the filter is size m, then I'm going to lose m minus 1 pixels. So the final size is going to be n minus m minus 1 in each direction. I'm assuming all receptive fields are square. Similarly, if I have a stride of S, the resultant value is going to be n minus m over S floored plus 1. This is just the numbers themselves are trivial enough for you to work out. The whole point is that this image is going to shrink even if you have a stride of 1. If you have a stride of more than 1, it's going to shrink by a factor. Okay. So moving on, sometimes you don't want the image to shrink for various reasons. Maybe you have the same code occurring again and again. Maybe you want to analyze all the way to the edges. In that case, what would you do? You want to make sure that the image doesn't shrink. So now, if I have n, an image of size n in each dimension, and if my filter is size m in each dimension, then I'm going to lose m minus 1 rows and m minus 1 columns. In order to not lose m minus 1 rows and m minus 1 columns, I'm going to pad zeros on either side. And the number of columns of zeros I'm going to pad is exactly going to be m minus 1, such that at the end of it all I get back a response as the same, which is the same size as the input. And the m minus 1 0 columns will be uniformly distributed on either side of the image, both left and right and top to bottom. If m minus 1 is an odd number, you'd have to choose which way you're going to distribute it, right? So anyway, why convolution? Actually, this I'm going to skip this call. Skip this. That was supposed to be a hidden slide. Now the convolution operation results in a convolution map, what we will call a convolution map. This is just the affine combinations. Subsequently, you must apply an activation, and an activation is applied to every entry in the map, and the activation, which just shows up in the pseudocode over here. Right after computing the z's, you apply an activation. That typically we will use ReLU as an activation, because as we saw, even when you go back to Fukushima's model, the ReLU kind of made biological sense. Subsequently, you're going to have, between convolutions, you have the downsampling layer, and the downsampling layer often alternate with convolutions, although this is strictly not necessary. So here's how the downsampling happens. Each downsampling layer is going to be looking at a collection of pixels or elements from a map. Now downsampling is not looking across all maps. Downsampling, again, the C cells, each C layer looks at only one S map, right? So that's going to be just one map. You'd be looking at a collection of values and retaining the largest one. Like here, if you had 3, 1, 4, and 6, you'd just be keeping the largest one, which is 6. And so you would slide left to right, and within each region, you're just going to keep the largest. Now, once again over here, till you got all the way to the bottom. Now, once again over here, it doesn't necessarily make sense to be sliding by one. In fact, for the downsampling layer, a stride of 1 doesn't make sense, because you picked the largest of those guys. Why should the same value contribute to 2 positions in the output image? So typically, the stride is going to be the same size as the region over which you're going to be taking the max. Now the point is, so over here, if you were trying to take a max pooling, the downsampling layer, over regions of size 2 cross 2. So the downsampling also has its little receptive field, right? Then this 4 cross 4 map is going to end up resulting in a 2 cross 2 map after the downsampling is performed. And typically for downsampling, you wouldn't zero pad because it doesn't make sense. Now, this is the biological aspect of it, right? But then now you can go beyond biology. There's nothing magical about taking the max. Why take the max? In the case of biology, in the case of the human eye, we were insistent on not missing things. The neural network doesn't have that issue. The neural network can do its own thing, right? So, which means that you can replace the max with a mean, and that makes total sense. The original max came from a completely different perspective. It came from a model of the human eye. But in computational terms, you can replace it with a mean. You could replace it with some kind of a p-norm. You can even replace it with a neural network, an MLP. which looks at those guys and decides what the layer must be. The whole point of this downsampling is to look at a combination of values and decide how to combine them to account for jitter for minor variations in position. And max is not the only operation you can perform. You can perform any operation. And sometimes you don't even need the downsample. In fact, turns out that the entire downsampling layer is not necessary. You don't need to follow biology. When you make an airplane, the airplane doesn't flap its wings. So you just need to maintain the concept. And that means you can just replace this whole downsampling with a regular convolution layer with a stride that's greater than 1. And that's going to reduce the size. And this is what is most commonly done these days. You don't bother with the downsampling. So here is the entire pseudocode. You can look it up because you'll need to code it, but this is without the downsampling. So here's the story so far. The convolutional neural network is a supervised version of a computational model of the mammalian brain. It includes conv layers and downsampling layers. Convolution can change the size of the output even for a stride of 1. This can be controlled by zero padding. Downsampling layers may perform maxes, p-norms, or be learned downsampling networks. Regular convolutions with a stride greater than 1 also result in a shrinking of the output and effectively perform downsampling. So let's put everything together for a typical image task. And I may just run about 5 minutes over, bear with me. The time taken in class times the size of the class is constant. Anyway, so let's consider this problem. I'm going to try to look for patterns in this color image to the left. But a color image actually comprises 3 images, an R, a G, and a B. Or if you're using some other key CMYK, it may have 4 planes, right? So then these R, G, and B can be stacked into a cube. Now, typically, this stuff is old. I'll skip this. So input, you have 3 pictures, right? Then I'm going to start off and say I'm going to have K1 total filters. I'm going to decide on how many filters, how many basic patterns I'm looking for in the input image. That is the number K1. And I'm going to be deciding on the size of these filters. So typically K1 is a power of 2, 2, 4, 6, 8. That's simply for computational convenience. And filter sizes tend to be small, 5 cross 5, 3 cross 3, or even 1 cross 1, because you want to be looking for really small features. If you make the features big, then there's too much variation possible within the feature. You're likely to lose some of them. So these are small enough to capture fine features, which is particularly important for scaled down images. Typically, if you get a 1000 cross 1000 image, you're not going to be working on a 1000 cross 1000 image. It's just going to take too much time. So you'd resample it down and you'd scale it down to something much smaller and tractable, say 128 cross 128, or even smaller, and work on those, which means that the size of the features are going to become really tiny, and you want to be really careful about not losing them. So the filters tend to be really small. Now, what is the strange business of a 1 cross 1 filter? That's really not looking at a pattern, right? It's not looking at a span. It turns out that's just simply a simple MLP, a simple perceptron. It's like which is looking at many inputs at the same time and producing many outputs at the same time. It's just one perceptron which is scanning, multi-output perceptron, which is scanning the image, right? So the input would be convolved with a set of K1 filters, each of a different size. So a better notation is going to actually show you the size of the filter and the number of channels, the number of planes. Here this is an RGB image. So the 5 cross 5 cross 3 tells you that each filter really requires 25 times 3, which is what? 125 parameters, because it's 3 deep. But I put the cross 3 in parentheses because you're only scanning across the X and Y. You're not scanning across colors, right? So the parameters to choose would be the number of filters, the size of the filters, and the stride itself. Now, you may or may not choose to zero pad, depending on whether you're willing to accept a reduction in the size of the image. So here is the computation that you're actually going to perform. Keep in mind that although I've shown the whole thing as a cube, it's actually many planes. So the computation consists of 2 steps. The first is where you compute the affine combination of the values in the 3 images over here. So you're going to call them. So you have several convolutional filters. The filters are 3D. The actual computation is going to be summing over RG and B, and within each RG and B you're going to be computing an inner product, which is a component-wise multiply of the filter values and the image values within a square region. You sum the lot, and you're going to get one such Z value for every position in a plane, and you're going to get several such planes, right? So once you get the Z values, you just apply an activation F on the Z's and you compute the value. The learnable parameters in the first layer, if I'm just working off images, are just going to be for each of the K1 filters, I'm going to require L cross L times 3 parameters. So the total number of parameters is going to be K times 3L squared plus 1. There's the bias, right? And the bias is because these are just neurons. You always have a bias. Now to answer the question of why a bias, if I'm looking at a max, I'm actually selecting the max value with the bias factored in. You're not storing the differential, you're storing the actual max value. So the bias is very much going to be part of the value that you actually compute. So the next operation is the pooling operation, which basically looks at little patches in the outputs of these planes and picks the largest value. So I could just write that as the Y's are the convolutional layers, the U's are the max pooling layers, and you can see that for each position in a U, now this each U is only looking at a square region in the corresponding plane, right? And you're just picking the largest value. You're literally just picking the largest value. Now when you actually implement this, the parameters to choose are the size of the pooling block and the pooling stride. Typically this pooling stride is going to be the same as the size of the pooling block itself, but sometimes it may not be for engineering purposes, right? There are other choices. Do you want to use max pooling? Do you want to use mean pooling? Or do you want to not use pooling at all? Just work with convolutions. Now when you actually implement it, you don't just say, so the equation that I wrote over here is this guy, right? Pick the largest of the values in the block. If you write it in this way, it turns out that implementing differentiation, remember you want to backpropagate derivatives. And when you backpropagate derivatives, just thinking of it as picking the largest value is not good for backpropagating derivatives. Now if you've gone through the slides of the previous lectures, you know that computing the derivative for a max operation really requires computing a subgradient, and which means that you have to retain the identity of the specific value that contributed to the max. A different way to think about it is, what is a derivative? I have 5 values, and if I'm picking a max, I'm asking the question, if I'm picking the max and then I compute a derivative, I'm asking a question, if I change this value a little bit, how much does the output change? So if this 5th value is the maximum guy that got chosen, then changing any of these other 4 values a little bit doesn't change the output. The only value which can be perturbed to change the output is the one that actually got chosen, which would be the 5th guy. So the derivatives for these 4 guys are going to be 0. The derivative for this guy is simply going to be 1, right? That was your max operation. So now in order for this derivative to be computed, you need to know which one was the actual max guy that got passed through. So the operation is going to be split into 2 steps. In the first step, within each block, you keep track of the position of the element that has the largest value. In the second step, you read the value from that position. So you compute the P, and from the P you compute the U, okay? And that is going to be the max pooling. And that's going to give you a bunch of planes, right? And then so some jargon, the filters are often called kernels. The outputs of the individual filters are called channels. If you have many filters, you're going to end up computing many planes, right? So each of these is called a channel. The number of filters is called the number of channels. This is just terminology. But basically the point is this: each filter will contribute a plane. Each of these planes we will call a channel. Each filter is going to be looking at many planes in the previous layer. So each filter is looking at many channels. To produce one channel, you're going to be producing a bunch of channels. That's just terminology, right? Then you can have a second convolutional layer. The second convolutional layer is going to have some K2 filters, K2 3D filters, each resulting in K2 two-dimensional maps, or alternately stating K2 channels. And each of those channels is computed K2 output channels. And each of the filters has K1 input channels. So you call them the number of input channels and number of output channels. All of these are various terminologies that people have come up with, and the terminology kind of proliferates. It gets a little more confusing with each day. But the basic concept is very straightforward, right? And so here, the parameters you'd have to choose is how many output channels, what is the size of the individual filters, and what is the stride that you would be skipping by when you're actually performing the convolution. So this is for the second layer of filters. The total number of parameters is going to be K2 because you have K2 output channels, right? For each of these output channels, the filter itself has K1, is looking at K1 input channels. Within each input channel, it's looking at a square of size L2, K1 L2 squared, and never forget the bias, right? The bias, this is just a standard perceptron. And so it's looking at a weighted combination of inputs plus a bias. So the total number of parameters has the extra +1, which is multiplied by the K2, right? Now, we may or may not use the bias, but conceptually the bias always exists. And from these K2 channels, you can perform max pooling. Take the size down. Here again, you'd have to choose the size of the max pooling block in the stride. And then finally, all of these, eventually after several layers of these, you're going to get one collection of maps, right, channels. And then there's an operation which we will call a flattening. This is just basically saying that I'm going to take this cube and I'm going to lay it out as a single vector. And then that vector can go onto a multi-layer perceptron. So, or it can just be typically you'll only use a single softmax. As we saw in the last class, the final multi-layer perceptron doesn't really make sense because when you're using a multi-layer perceptron, this is like the equivalent of scanning without sharing parameters. And that's a very inefficient thing to be doing. So typically we'll only just use a final softmax. Now, each convolution layer maintains the size of the image modulo the boundary effects. Each convolution layer may increase the number of maps, increase the number of channels. So you may start off with 3 channels and end up with 27, right? The first layer, for example, has the number of input channels is 3, R, G, and B. The number of output channels can be anything. It depends on how many neurons, how many filters you have for the first layer. If you have 100 filters, then it's going to the number of output channels is 100. So you can start off with a cube which has 3 planes and end up with a cube which has 100 planes, right? Each pooling layer with a hop D decreases the size of these maps. The number of planes remains the same. The area decreases, and so that downsampling is going to reduce the size of the images. The various parameters you choose, number of convolutional and downsampling layers, the arrangement, where you would use the convolution layers, if downsampling layers, if any. In each layer, you're going to look at the number of filters, the extent, and the stride. For downsampling layers, you're going to be looking at the extent and the stride. And for the final MLP, all the traditional parameters of an MLP, right? So training is going to be done exactly as in a regular MLP. The only difference is in the structure of the network. So you would be providing training examples of images and classes. You define a divergence between the desired output and the true output of the network in response to any input. And then network parameters are trained through variance of gradient descent. Gradients are obtained through backpropagation. So the parameters to be learned are all of these guys, the weights and biases of the neurons in the final MLP, and the weights and biases of the filters for every convolutional layer, right? So I'm actually going to, I'll stop right here, but the actual training procedure itself we will go through in the next class. If you have any questions, you can post on Piazza, or maybe we have a minute to ask questions. We actually have 5 minutes. Let me finish the lecture, right? So, which is 2 more slides. So in the final flat multi-layer perceptron, all of the weights and biases must be learned, right? In the convolutional layers, all of the filters must be learned. So in each layer J, we have KJ maps, right? KJ output channels, basically. And each output channel is being computed over a region of some Lj cross Lj times Kj minus 1, right? And the plus 1 bias for the bias. So the total for the jth layer, this is how many parameters, filter parameters you'd have to estimate within the convolutional layers. So this is in addition to the MLP itself. And the loss, you have a final output from the MLP. This is just the usual softmax output. The loss would be defined in much the same manner that you would define the loss for any traditional MLP. Now remember that although this entire network looks very complicated, this is just one giant multi-layer perceptron, right? We saw this in the last class. Scanning with an MLP is the same as composing one large MLP, with the distinction that this large MLP has shared parameters, right? So that's about all that happened. And so the learning rules are going to be exactly the same as those for a regular MLP, except that now you would have to account for the fact that parameters are shared. And then this can result in some kind of efficiencies more than anything else. It doesn't really change the basic concept. And so the problem setup is you'll be given a bunch of training instances. On the ith training instance, you can define the divergence. You have the total loss, which is the average divergence across all of your training instances. And you want to minimize this loss with respect to all the weights and biases for every single perceptron, every single neuron in your network, which includes the neurons in the final MLP and the individual filters, where each filter eventually we saw is just a regular neuron, right? So although we may call them filters, we may call them input channels, we may call them output channels or whatever else, basically each filter is just a neuron, right? All of these must be learned. So here's the training loss. Here's this very high level pseudocode. The gradient descent algorithm, you would be going through all of the layers iteratively and just use the standard gradient descent rule to learn the parameters till the loss is converged. The real key is how do you actually compute this derivative, right? And that derivative, this is just the derivative of the loss, which is the average divergence. So it's just the average of the divergences, derivatives of the divergences for the individual training instances. Nothing has changed. Absolutely nothing has changed. And this is the key term that we must figure out how to compute. And we'll do that in the next class, right? So you can just use backpropagation continuous in through the MLP in the usual manner until you get to the first flat layer, which is just the unrolling of the final convolution layer, and then it's going to travel backwards from there. Here we will need some adjustments to account for the fact that these are shared parameter networks. And we'll see how we actually adjust this in the next class. Questions? Thank you.